There are many ways to die in the world: cancer, motor vehicle accidents, or even getting smashed alive by a vending machine. Maybe surprisingly, the most common cause of death in the world isn't any of these. It's actually cardiovascular disease, and we're here to talk about atherosclerosis, which is a leading cause. Atherosclerosis is a condition that involves the buildup of lipid in the arteries, and related complications include heart attacks and strokes and are responsible for one-third of all deaths annually. Often, the first symptoms of atherosclerosis are fatal, so it's important that we're able to predict the disease's progression. Current predictive models mainly focus on clinical parameters, but we aim in this project to increase model accuracy by taking gene expression into account as well.
So we used a data set containing 391 human blood samples split into cohorts based on disease severity level to make these models. And these severity levels are control, mild, medium, and severe atherosclerotic disease. And we split this data set into a binomial and multinomial disease severity labeling. In the binomial data set, we set the control as no atherosclerosis, and we set the other three levels as yes atherosclerosis. And compared to the multinomial data set, where we set just four of the levels as their own categories. And we randomly split both the binomial and multinomial data sets into a random selection of 80% training data and 20% testing data. And we developed 6 classical machine learning models off of these using bootstrapping, which accounts for a lack of testing data, and stratified shuffle split, which makes for a random selection for even distribution between training and testing data. And from those 6 models, the binomial, logistic regression, and support vector machine models produced the highest accuracy and precision recall AUC scores. The SVM model produced a ROC AUC score of 0.74 and a precision recall score of 0.83, which outperforms some models that only include clinical factors, models such as the Kirk study. And we also found overlaps in selected genes that were found to be positively correlated with atherosclerosis from our models were also found in other studies to be upregulated during atherosclerosis, and vice versa.
In the future, we hope to refine and train this model, hopefully with more data sets and with those data sets having more diverse patient samples. And we will prioritize the binomial logistic regression model and support vector machine models when we do this. Thank you.
Thank you.